Antimicrobial Chemotherapy.
And before we get into the biochemistry of all that, I want to pause on that word chemotherapy, because if I say that to you or to someone on the street, you immediately think of cancer. You think of hair loss, nausea, radiation rooms. But that's not really what the word means in its purest sense, is it?
No, not at all. Chemotherapy simply means the use of chemical agents to treat a disease. It could be cancer, yes, but taking an aspirin for a headache is technically chemotherapy. In our context today, though, we are looking at this specific arsenal of drugs designed to kill microorganisms, bacteria, mostly inside a living human body.
And that brings us to the central tension of this entire deep dive. The sources keep coming back to this one phrase, selective toxicity. It sounds like an oxymoron. How can something be toxic, which means poisonous, but also selective?
That is the holy grail. Think about it this way. If you have a bacterial infection in your throat, I could easily kill those bacteria. I could have you drink a glass of bleach. The bacteria would die instantly.
But so would I, which isn't ideal.
Exactly. That is toxicity, but it's not selective. The challenge of antimicrobial medicine is to find a magic bullet, a poison that acts like a sniper shot. It has to identify a target that exists only on the bacteria and not on the human host. It has to kill the invader while leaving you completely unharmed.
And finding those targets, those biological differences between us and them, is essentially the map for today's discussion. But let's rewind the clock first, because for most of human history, if you got a scratch that got infected or you contracted pneumonia, you were essentially rolling the dice with your life.
You really were. We had very few tools. We knew about quinine from cinchona bark for malaria, and we knew about emmetine for amoebic dysentery, but those are for parasites. For bacteria, nothing. The real scientific approach didn't start until the early 20th century with Paul Ehrlich.
The man who coined the term magic bullet. I was reading about his work with syphilis. He wasn't playing around with gentle herbs. He was using arsenic.
He was. He developed the arsenopamines. It was the first planned chemotherapy. He chemically modified arsenic to try and make it less toxic to humans, but still lethal to the syphilis spirochete. It worked, but it was dangerous. It was a very messy, very toxic start.
Then we hit the 1930s, Gerhard Domack. This story always fascinates me because it starts with dye, right?
Crontosil, yes, a red dye. Domack found that this dye could protect mice from streptococci. It was the birth of the sulfonamides. But the interesting thing is, Prontosil itself isn't active in a test tube. It only works inside the living body.
Why is that? Why wouldn't it work in a petri dish?
Because the body's metabolism actually breaks the dye down into the active ingredient, which is sulfonilamide. It's a massive breakthrough. But as big as that was, it was about to be completely overshadowed by the discovery that really defined the century.
The mold juice. Penicillin.
Alexander Fleming, 1929. The story is legendary. He leaves a petri dish out, a mold spore lands on it, and he notices that the bacteria around the mold are dissolving. But here's the part people usually forget. Fleming didn't turn it into a medicine.
Right, there's this huge gap in the timeline. He publishes a paper, says, hey, this is interesting, and then nothing happens for a decade.
Almost nothing, yeah. It was incredibly difficult to purify, it was unstable, and people just didn't grasp the clinical potential. It wasn't until 1940, driven by the absolute desperation of World War II, that Howard Florey and Ernst Chain at Oxford really turned it into a therapeutic substance. And once they did, the floodgates opened.
The golden age of antibiotics.
From 1940 to about 1965, we discovered almost every major class of antibiotic we use today. And we didn't invent them in a lab so much as we found them. We went out to the soil, found bacteria and fungi that were fighting each other for territory, and we essentially stole their weapons.
So today, we are going to deconstruct that exact arsenal. We have a massive amount of material from the sources to get through. We need to break this down by mechanism. How do these drugs actually kill?
We can group them into four main strategies. 1, destroy the wall. 2, pop the membrane. 3, sabotage the factory, which is protein synthesis. And 4, crypt the code, the DNA and RNA.
Let's start with the first one, which seems the most intuitive, targeting the wall. Why is the bacterial cell wall such a perfect target for selective toxicity?
Because you don't have one.
I mean, I feel like I hit a wall sometimes, usually around 3 p.m. But biologically?
Biologically, your cells are held together by a flexible cell membrane. But bacteria, they are under immense pressure. The osmotic pressure inside a bacterium is incredibly high. If they didn't have a rigid mesh-like armor around them, they would literally explode just from existing in the watery environment of your body.
So the wall is like the pressurized hull of a submarine.
Exactly. And this wall is made of a unique stuff called peptidoglycan. Imagine a chain link fence. You have long chains of sugars, specifically N-acetylglucosamine and N-acetylmuramic acid. We call them NNM.
Okay, so these NAG and NMM sugars are the long horizontal wires of the fence.
Right. But a fence isn't strong unless you clip the wires together vertically. The bacteria use short chains of amino acids to cross-link these sugar strands. The enzyme that does this, the builder that clips the fence together, is called a transpeptidase. It's also known as a penicillin-binding protein, or PBP.
And this is where the drug strikes.
This is the kill shot. Beta-lactam drugs, like penicillin, look structurally similar to the normal building blocks. They trick the transpeptidase enzyme. The enzyme grabs the penicillin thinking it's a piece of the wall it needs to attach, and the penicillin binds to it irreversibly. It completely jams the zipper.
So the builder is stuck holding the drug and can't do any more work.
The cross-linking stops. But the bacteria keeps growing. It keeps producing stuff inside. The pressure builds. Not only that, but jamming the enzyme triggers autolytic enzymes. The bacteria basically turns on its own self-destruct mechanism. The wall gets weaker and weaker until, boom, lysis. The bacteria bursts open.
That is visceral. So when we say beta-lactam, we're referring to the specific chemical ring shape that jams this lock.
Correct. The beta-lactam ring is the actual weapon. But the bacteria aren't passive victims. They fight back. They produce enzymes called beta-lactamases. Think of these as molecular scissors. They float around, find the penicillin molecule, and snip that beta-lactam ring open before it can ever reach the target.
And this back and forth, we make a drug, they make a scissor, we make a better drug, is basically the entire history of the penicillin family.
Ideally, yes. Let's look at the penicillins to see how that played out. You have the originals, penicillin G and penicillin V. These are still great for simple bugs like strep, pneumococci, meningococci. But they have a weakness. Penicillin G is destroyed by stomach acid. You have to inject it IV or IM. Penicillin V was modified to survive the stomach, so that's the oral version.
But then Staph aureus figured them out pretty quickly.
Staph is smart. It started producing penicillinase, which is a specific type of beta-lactamase. So plain old penicillin simply stopped working on staph. We had to escalate. We developed the penicillinase-resistant group, also known as the anti-staph penicillins. Drugs like nafcillin, oxacillin, and methicillin.
How do they work? Did we change the weaponized ring?
We kept the ring, but we added bulky chemical groups around it. Imagine putting a cage around the key. The bacterial scissors, the beta-lactamase, couldn't physically fit in to cut the ring because the bulky groups were in the way.
So the drug survives and kills the staph. But we've all heard of MRSA, methicillin-resistant Staphylococcus aureus. If methicillin was armored against the scissors, how did the bacteria beat it?
This is where the evolutionary story gets scary. MRSA didn't build better scissors. It changed the lock entirely. It altered the shape of its transpeptidase enzyme, the PBP. Now, the methicillin molecule, armored or not, simply doesn't fit into the builder enzyme anymore.
That is a fundamental shift. It's not attacking the drug. It's just completely ignoring it.
Exactly, and that renders that entire class of drugs completely useless against that strain.
Okay, so we have the anti-staph group. Then we have the broad-spectrum group ampicillin and amoxicillin. I feel like everyone listening has taken amoxicillin at some point in their lives.
They are the absolute workhorses of medicine. They were designed to move better through the pores of gram-negative bacteria. Gram-negatives are tricky because they have an extra outer membrane that acts like a bouncer. Ampicillin and amoxicillin can slip past the bouncer better than regular penicillin.
But they are vulnerable to the scissors, right? That's why you so often see amoxicillin paired with something else, clavulanic acid.
This is one of my favorite strategies in pharmacology. Clavulanic acid isn't really an antibiotic. I mean, it doesn't kill bacteria well on its own at all. It's what we call a suicide inhibitor.
A suicide inhibitor, that sounds dramatic.
It looks like a beta-lactam. It dives onto the bacterial beta-lactamase scissors and binds to them permanently. It literally sacrifices itself. It keeps the scissors busy so that the amoxicillin can sneak through, find the builder enzyme, and stop the wall construction.
That is incredible teamwork. It's the Secret Service agent jumping in front of the bullet.
It really is. And finally, in the penicillin world, we have the anti-pseudomonal drugs like piperacillin and ticarcillin. Pseudomonas is a nasty water-loving bug often found in hospitals. It has very tight pores and it actively pumps drugs out. Piperacillin is the heavy hitter designed specifically to penetrate those tight defenses.
All right, let's slide over to the cousins of penicillin. The cephalosporins. I see these listed in generations, first through fourth, sometimes fifth. Is this just marketing, like with smartphones?
It's more like a timeline of tactical needs. They all share a slightly different core structure, a 7-imidocephalosporanic acid nucleus. But as we move from the 1960s to the 90s, the clinical problems changed. The first generation, drugs like cefacillin, are very much like penicillin. They are great at killing gram-positive bugs like staph and strep. We use them all the time to prevent infection during clean surgeries.
But they struggled with the gram-negatives.
Right. So the second generation was developed to increase that gram-negative coverage. But the real leap, the game changer, was the third generation, drugs like ceftriaxone and ceftazidime.
What made the third generation so special?
Two things. First, they are excellent at killing gram-negatives. Second, and this is crucial for saving lives, they can cross the blood-brain barrier. If you have bacterial meningitis, an infection of the covering of the brain and spinal cord regulator, penicillin doesn't get in there very well. Ceftriaxone walks right in.
That explains why it's the absolute go-to for meningitis. And the fourth generation.
Cefepime. It's the best of both worlds. It hits gram-positives hard, it hits gram-negatives hard, and it is much more resistant to those scissor enzymes. We also have newer ones, like ceftaroline, which is specifically engineered to bind to the mutated lock in MRSA.
Before we leave the beta-lactams entirely, we have to talk about what the sources call the nuclear option, the carbapenems, imipenem, meropenem.
We call these the broadest spectrum antibiotics we have. Their structure is just different enough that almost no standard beta-lactamase can cut them. When a patient is dying of severe sepsis in the ICU and we don't know what the bug is, we often reach for these because they wipe out almost everything.
But there's a really weird footnote in the text here about imipenem. It needs a sidekick called cilastatin. Is that another suicide inhibitor for bacterial resistance?
Surprisingly, no. That's for human resistance. It turns out there's an enzyme in the human kidney called dehydropeptidase that breaks down imipenem naturally. If you just gave the drug alone, the patient's kidneys would destroy it before it could even circulate and work. Cilastatin is added to block that kidney enzyme.
So we are chemically hacking our own kidneys to let the drug do its job. That's wild. Now, there are a couple more things here. Monobactams?
Aztreonam. It's a beta-lactam, but the ring is completely standalone. It only works on aerobic gram-negative rods. The real beauty of Aztreonam is that if a patient has a severe anaphylactic allergy to penicillin, you can usually give them this safely because the structure is so different.
Good to know. Now, there is one more major wall destroyer that is definitely not a beta-lactam, vancomycin.
The tank. Vancomycin is a glycopeptide. It is a massive, heavy molecule. It works on the cell wall, but not by jamming the builder enzyme like penicillin does.
How does it work, then, if it's not jamming the lock?
Remember the chain link fence analogy? The amino acid chains that get clipped together end in two specific molecules, D-alanine, D-alanine. The builder enzyme needs to grab those two molecules to make the clip. Vancomycin comes in and literally sits on top of the D-al end. It wraps it in a giant bear hug.
So it's physically covering the docking port.
Exactly. The builder enzyme comes along, looks for the D-al to do its job, but can't find it because vancomycin is sitting on it. Construction halts. Lysis follows.
But because it's a tank, a huge molecule, it has limitations.
Big time. It physically cannot fit through the tiny pores of the outer membrane in gram-negative bacteria. It's just too big. So vancomycin is strictly for gram-positives. It has been the gold standard for treating MRSA for decades.
But even the tank is starting to fail. The sources mention VRE vancomycin-resistant enterococci.
And the mechanism here is elegant and terrifying. The bacteria mutated at the very end of the chain. Instead of putting D-alanine at the end, they started making D-alanine D-lactate.
Just one molecule difference, alanine to lactate.
Just one. But it changed the molecular shape just enough that vancomycin can't hug it anymore. It slips off. The builder enzyme can still use the lactate version to cross-link the wall. So the wall gets built and the bacteria survives.
It's a constant game of cat and mouse.
Mm.
Oh, and before we move on, bacitracin is also listed here as a wall inhibitor.
Yes, but bacitracin is highly toxic if taken systemically.
Yeah.
It ruins the kidneys. So we only use it topically. If you put antibiotic ointment on a cut, like Neosporin, that's bacitracin at work, safely outside the body.
All right, let's move to the second mechanism. If you can't break the wall, destroy the skin beneath it, the cell membrane.
This is a much cruder method. The cell membrane is a lipid bilayer. It's grease and oil, basically, the permeability barrier. The drugs we use here, the polymyxins, are essentially detergents.
Like dish soap.
Very similar principle. They are basic cationic polypeptides. They have a positive charge that is strongly attracted to the negative charge of the bacterial membrane. Yeah. They insert themselves into the membrane and disrupt the structure. They literally poke holes in it. The insides leak out and the cell dies.
But here's the problem with that. Human cells have lipid membranes too. We don't have walls, but we definitely have
We
membranes.
do. And that's exactly why polymyxins are so toxic. They can damage our kidney membranes, causing nephrotoxicity, and our nerve membranes, causing neurotoxicity. Historically, we stopped using them systemically because they were just too dangerous. We relegated them to topical creams.
But the sources say they are making a comeback in hospitals.
Only out of sheer desperation. For multi-drug resistant gram-negatives like Acinetobacter or Pseudomonas that laugh at every other drug we have, we bring out the polymyxins, specifically colistin. It's a very calculated risk. We know we might hurt your kidneys, but if we don't use it, the bacteria will kill you.
There's another membrane drug mentioned here, daptomycin.
A cyclic lipopeptide. It works a bit like a taser. It inserts into the cell membrane of gram-positive bacteria in a calcium-dependent manner, and it causes rapid depolarization. It essentially drains the electrical battery of the cell. Without that electrical potential, the cell can't make DNA, RNA, or protein. No electricity, no life. It's great for VRE and MRSA.
But it has a quirky side effect regarding muscles.
It can cause myopathy and even rhabdomyolysis, which is the breakdown of muscle tissue. So if you are on daptomycin, we have to monitor your blood for an enzyme called CPK to make sure your muscles aren't dissolving.
Okay, so we've breached the wall, we've dissolved the membrane. Now let's go deep inside the castle. Mechanism 3, sabotaging the factory, inhibition of protein synthesis.
This relies on a very specific, beautiful structural difference. Both humans and bacteria need ribosomes to build proteins from genetic instructions. But our ribosomes are different sizes.
Right. The sources say humans have the 80S ribosome and bacteria have the 70S.
Exactly. And the bacterial 70S is made of two subunits, the 30S subunit and the 50S subunit. It's like a complex typewriter with two main gears. Because of this size difference, we achieve selective toxicity. If we can jam a wrench into specifically the 70S gears, we can stop the bacteria from making proteins without stopping the human host's protein production.
Let's look at the wrench that hits the smaller gear first, the 30S subunit, the aminoglycosides. Drugs like streptomycin, gentamicin, and amikacin.
These are old, incredibly powerful drugs. They bind to the 30S subunit and do something very specific. They freeze the initiation complex, and they cause the ribosome to misread the messenger RNA.
So the RNA blueprint says, make protein A, but the ribosome stutters and makes protein garbage.
Exactly. The bacterium floods itself with dysfunctional, often toxic proteins. It breaks up the polysomes into non-functional monosomes. This kills the cell outright. That's why aminoglycosides are bactericidal, they kill. Most other protein synthesis inhibitors just stop growth, making them bacteriostatic. These guys kill.
But again, there's a massive cost. These drugs are notorious for side effects.
They are highly ototoxic. They accumulate in the fluid of the inner ear and literally kill the hair cells that help you hear and balance. And it's often irreversible deafness. They're also highly nephrotoxic, shutting down the kidneys. We have to draw blood constantly to monitor the peak and trough levels of the drug to keep you safe. Oh, and they're often used in synergy with penicillin. The penicillin breaks the wall, allowing the aminoglycoside to flood in and hit the ribosomes.
Synergy, I like that. Then we have the tetracyclines, doxycycline, minocycline.
These also hit the 30S subunit, but they are gentler. They just block the docking bay where the raw materials, the transfer RNA, come in. They just stop the factory line from moving forward. They are bacteriostatic.
And the side effect warning here is very specific for kids and pregnant women. Why is that?
Tetracyclines absolutely love calcium. They are chili odors. If a fetus is developing bones in utero or a young child is growing their adult teeth, the drug binds to the circulating calcium and gets deposited directly into the bone or tooth matrix. It causes a permanent gray or brown discoloration of the teeth and can even stunt bone growth.
So no doxycycline for the little ones. The text also mentions glacocyclines.
Tiazocycline. It's basically a synthetic derivative of tetracycline designed to beat resistance. It binds much tighter to the 30S subunit, so it works on MRSA and VRE.
Let's move to the bigger gear, the 50S subunit.
We have a few major classes here. The macrolides, erythromycin, azithromycin, which everyone knows is the Z-Pak and clarithromycin. They bind to the 50S and block translocation. Basically, they block the tunnel where the newly formed protein chain tries to exit the ribosome. It's like clogging the exhaust pipe. They are great for community-acquired pneumonia, Legionella, chlamydia.
Then there's chloramphenicol. The notes say it causes gray baby syndrome. That sounds like a horror movie title.
It's a tragic reality of human metabolism. Chloramphenicol blocks peptidyltransferase on the 50S. It's a very effective drug. But it is processed and detoxified by the liver using an enzyme called glucuronyltransferase. Newborn babies, especially premature ones, simply haven't developed this enzyme yet.
So they can't clear the drug from their system.
Right. The drug builds up to incredibly toxic levels, causing cardiovascular collapse.
Right.
The baby turns an ashen, cyanotic gray color and goes into shock.
Yeah.
It also causes severe bone marrow suppression in adults. That's why we almost never use it in the developed world anymore, except for extreme life-threatening cases like severe Rocky Mountain spotted fever or meningitis where nothing else works.
And clindamycin.
That's a lincosamide.
Right.
Also hits the 50S. It's great for anaerobic bacteria, but it's infamous for what it does to the gut microbiome. It kills the normal flora so effectively that it allows Clostridium difficile C. diff to take over. It's the #1 classic cause of antibiotic-associated pseudomembranous colitis.
It fixes one problem and starts a much worse one.
Yep.
Finally, in the factory sabotage department, we have the oxazolidinones, linezolid.
The relatively new kid on the block. It has a totally unique mechanism. Instead of jamming a working ribosome, it stops the ribosome from even assembling in the first place. It prevents the 30S and 50S subunits from clicking together to form the initiation complex. Because it's a completely novel mechanism, there wasn't any pre-existing cross-resistance. It works beautifully on the superbugs MRSA and VRE.
All right, section 4, we've done the wall, the membrane, the factory. Now we go for the blueprints themselves. Inhibition of nucleic acid synthesis.
We can target the DNA or the RNA. Let's start with DNA. The quinolones or fluoroquinolones, ciprofloxacin, levofloxacin.
Cipro, a total household name. How does it break DNA?
It targets an enzyme called DNA gyrase, also known as topoisomerase X and topoisomerase IV. Think about a phone cord, the old coiled ones. If you pull apart the 2 strands of the DNA double helix to read it or copy it, the rest of the coil gets overly knotted and tight. It supercoils.
And if it gets too tight, it snaps.
Right. DNA gyrase is the bacterial enzyme that continuously cuts, untwists, and reseals the DNA to relieve that tension ahead of the replication fork.
And the quinolones jam that specific enzyme.
They freeze it while the DNA is cut. The DNA strands are severed, the tension is catastrophic, and the bacteria dies.
But there's a bizarre side effect with Cipro involving tendons.
Yes, tendinitis and even spontaneous tendon rupture,
Yes.
specifically the Achilles tendon. It's a black box warning. We think it's because the drug might affect human collagen synthesis, or perhaps it chelates magnesium in the tendon tissue, weakening it. It's rare, but if you're a runner on Cipro, you really need to be careful and avoid strenuous exercise.
And targeting RNA.
The rifamycins. Rifapin is the main one. It binds strongly to the bacterial DNA-dependent RNA polymerase. That's the machine that reads the DNA and transcribes it into messenger RNA. It jams the machine. No RNA means no proteins.
This is a key drug for tuberculosis, right?
Absolutely essential for TB. But it has a very famous clinical quirk. The drug itself is a highly pigmented, reddish-orange compound, and it dyes all of your body fluids. Your urine, your sweat, your tears, they all turn bright orange-red.
Which is harmless, but totally terrifying if you aren't expecting it.
Imagine sweating red or crying orange tears and ruining your contact lenses. You absolutely have to warn the patient before they leave the clinic, or they will end up in the ER thinking they're bleeding out of their eyes.
Finally, under this nucleic acid section, we have the metabolic inhibitors, the folic acid pathway, the sulfonamides and trimethoprim. This brings us back to that selective toxicity idea in a really beautiful way.
It's the most elegant mechanism we have. It's all about folic acid. Every single cell needs folic acid to synthesize purines and pyrimidines, the building blocks of DNA. Humans, we simply eat it. We get our preformed folate from leafy greens and vitamins. We have transport mechanisms to bring it into our cells. Bacteria, they are completely impermeable to external folic acid. They must manufacture it from scratch, inside the cell.
So they have a mandatory factory line to build folic acid and we don't?
Exactly. So if we block that factory line, the bacteria starve and can't make DNA, but the human host is completely unaffected because we just get our folate from a salad.
And this is where the synergy comes in. We almost always give a sulfa drug and trimethoprim together, like in Bactrim. Why?
It's called a sequential block. The factory line has multiple steps. The sulfa drug looks exactly like the first raw material, which is called pabia. It acts as competitive inhibitor, jamming the first enzyme, dihydroapatite synthase. But some bacteria can push past that. So trimethoprim is designed to resemble the folate molecule later in the process. It jams the next key enzyme in the chain, dihydrofolate reductase.
So if a bacterium manages to slip past the first roadblock, the second one catches it.
Right, and here is the magic. While each drug alone just pauses growth, they are bacteriostatic. When you use them together, they shut down DNA production so completely that they become bactericidal. They kill the bug.
That covers the arsenal. But now we have to talk about the enemy's response. Section 5, the Empire Strikes Back, resistance.
Bacteria are the ultimate survivors. They reproduce every 20 minutes. Evolution happens in fast forward.
The sources mention non-genetic resistance first, like persisters. What are those?
Persisters are metabolically inactive bugs. Remember, penicillin only works if the bacteria is actively building a wall. If a few bacteria in a colony just stop growing and go dormant, the antibiotic does nothing to them. Once the drug is gone, they wake up and start dividing again. There's also location. Sometimes the drug just physically can't reach the bug because it's walled off in an abscess or hiding inside a human macrophage.
But the real threat is genetic resistance. We talked about chromosomal mutations like the MRSA target change. But what fascinates me is the transferable resistance, extrachromosomal, plasmids.
This is the nightmare scenario for infectious disease doctors. A plasmid is a small circular loop of DNA that exists separate from the main bacterial chromosome. Think of it like a USB flash drive.
Wow.
And on that drive, the bacteria stores cheat codes for survival, resistance genes.
And they can share these drives with each other?
Readily. The main method is conjugation. Two bacteria, and they don't even have to be of the same species, can link up via a tube called a sex pilus. One copies its resistance plasmid and shoots it across the bridge into the other. So a harmless gut bacteria can learn how to destroy penicillin, pass that flash drive to a dangerous salmonella passing through, and suddenly you have an untreatable outbreak.
What exactly is coded on these flash drives? We mentioned the beta-lactamase scissors. What else?
We group them into 5 how-to methods of resistance. 1. Make an enzyme to destroy the drug, like the scissors. 2. Alter the target, like MRSA, changing its lock so the drug doesn't fit. 3. Change permeability, just close the outer membrane pores so the drug literally can't get inside. 4. Create an altered metabolic pathway. If we block one road to make folic acid, they evolve a detour around the block.
And the 5th one?
Efflux pumps. This is fascinating. Purely mechanical resistance. The bacteria builds a pump in its cell membrane that actively transports the antibiotic out of the cell. The drug gets in, and the pump grabs it and uses ATP to spit it back out before it can find its target. Tetracyclines and macrolides often fail because of these pumps.
It's literally bailing water out of a sinking boat.
Exactly. And the pumps can sometimes bail out multiple classes of drugs at once, creating multi-drug resistance instantly.
Moving to section 6, the clinical battlefield. Because it's one thing to understand the chemistry in a lab, it's another to actually cure a patient.
Right. In the lab, in vitro, we determine the MIC, the minimum inhibitory concentration. We put the bug in a test tube and see exactly how much drug is needed to stop it from growing. We also check the MBC, the minimum bactericidal concentration, to see what it takes to kill it. But the human body is not a controlled test tube.
We have absorption issues, stomach acid, liver metabolism, kidney excretion.
And distribution. The drug has to reach the site of infection in a high enough concentration. If you have an infecting deep in the bone, osteomyelitis, can the drug penetrate the dense bone matrix? If you have meningitis, can the molecule physically cross the blood-brain barrier? Many drugs can't.
And we can't forget the host's immune system, which the sources highlight as a critical variable.
It is the most critical variable. People assume antibiotics do all the work. They rarely do. They usually just knock the bacterial numbers down to a manageable level so your white blood cells can sweep in and finish the job. But if you are immunocompromised, say you have AIDS or you are on chemotherapy for cancer or you're an organ transplant patient, we can't rely on bacteriostatic drugs that just pause growth. Your immune system won't clean up the sleeping bugs. We absolutely need bactericidal drugs that kill the bacteria outright.
There's also the issue of collateral damage, the microbiota.
Right, the normal flora. Your gut, your skin, your mouth are covered in trillions of good bacteria that compete for space and keep pathogens out. When you take a broad-spectrum antibiotic, it's like carpet bombing. You kill the good guys along with the bad. This leaves wide open ecological niches.
Which leads to superinfections.
Exactly.
Yeah.
Yeast infections caused by Candida are incredibly common after antibiotics. Or, as we mentioned earlier, the dreaded C. diff colitis. Or sometimes natural resistant bugs like Proteus or Pseudomonas take over the gut.
Strategy and selection. Section 7. A patient walks into the ER with a high fever, a stiff neck, and confusion. You suspect bacterial meningitis, you do a lumbar puncture, take a sample. But the lab takes 48 hours to grow the culture and test the MIC. You clearly can't wait 2 days to treat them. What do?
We use empiric therapy, the best guess. Based on the patient's age, their history, and the symptoms, you know the statistically likely pathogens. For meningitis in an adult, it's probably pneumococcus or meningococcus. So you hit them hard immediately with a broad-spectrum cocktail, maybe ceftriaxone, to cross the blood-brain barrier, plus vancomycin, just in case it's a highly resistant strep. You cover all the likely bases.
And then 2 days later, the lab calls, hey, it's pneumococcus, and we tested it. It's highly sensitive to plain old penicillin.
Then you must de-escalate. You stop the big guns, you switch them to narrow-spectrum penicillin G. This reduces the toxicity to the patient, it saves money, and crucially, it stops the selective pressure that forces your normal flora to develop resistance.
We also mentioned combination therapy earlier, using 2 drugs at once, but there is a huge warning in the text about antagonism.
This is a classic pharmacological trap. Imagine you mix penicillin with tetracycline. You think 2 drugs are better than 1, right?
But penicillin only works if the bacteria is actively growing and building its cell wall.
Exactly. And what does tetracycline do? It's a bacteriostatic protein synthesis inhibitor. It stops the bacteria from growing.
Ah, I see. The tetracycline freezes the bacteria, the penicillin floats by, looks at the bacteria and says, well, they aren't building walls right now, so I can't do anything.
You have completely neutralized your own weapon. The bacteriostatic drug antagonized the bactericidal drug. You have to be very careful about understanding the mechanisms before you mix them.
What about prophylaxis? Using these drugs to prevent an infection before it starts.
We do it, but we have to be judicious. The best examples are giving a dose of cefazolin right before making the incision in a clean surgery to kill any skin staph that gets in. Or if a patient has a prosthetic heart valve, we give them amoxicillin before they go to the dentist.
Why the dentist?
Because dental cleanings push mouth bacteria into the bloodstream. Normally, your immune system clears it in minutes. But if you have a fake heart valve, the bacteria can stick to the metal or plastic and form a deadly vegetation. The prophylactic antibiotic kills them before they can stick. But generally, giving antibiotics to healthy people just in case is a terrible idea that drives resistance.
Let's wrap up with the special arsenals in Section 8. Tuberculosis. Why is TB so uniquely hard to treat? Why does it take 6 to 9 months of continuous pills?
TB is a fortress. The bacteria, Mycobacterium tuberculosis, grows very, very slowly. As we learned, many antibiotics only work on fast-dividing cells. Second, it hides intracellularly, living right inside our own immune macrophages. And third, it has a thick, waxy cell wall made of mycolic acid that physically repels most normal drugs.
So we use a very specific cocktail. RIPE therapy. Rifampin, isoniazid, pyrazinamide, and ethambutol.
We have to use all 4 drugs at once initially. The mutation rate for TB is such that if you use only 1 or 2 drugs, the bacteria will inevitably develop resistance and kill the patient. Isoniazid, or INH, is the backbone. It specifically inhibits the synthesis of that waxy mycolic acid wall. But it has a catch. It depletes vitamin B6 pyridoxine in the human body, which causes peripheral neuropathy nerve damage. So we always have to prescribe a B6 supplement alongside it.
And ethambutol has that weird side effect on the eyes?
Optic neuritis. Patients could lose visual acuity and lose the ability to distinguish between red and green. It's usually reversible if you stop the drug promptly, but you have to test their vision regularly while they're on it. Pyrazinamide works best in the acidic environment inside the macrophage where the TB hides, and rifampin, as we discussed, stops the RNA.
What about simple, uncomplicated UTIs, urinary tract infections?
Nitrofurantoin is a fascinating drug for that. It's an antiseptic, really. It gets absorbed, but it is excreted into the urine so rapidly that it doesn't achieve high enough levels in the blood to have systemic effects. It essentially just turns your bladder and your urine into a localized disinfectant tank.
And lastly, the anaerobes, the bugs that live in the deep, oxygen-free parts of the gut or inside deep puncture wounds.
Metronidazole. It's the ultimate anaerobic specialist. It works by generating highly reactive free radicals inside the bacteria that completely shred their DNA. But it only generates those radicals in the absence of oxygen, so it spares human cells and aerobic bacteria. It's the go-to for bacteroids in the gut, or Clostridium. And weirdly, it also treats protozoal infections like Giardia from drinking bad stream water.
But there is a massive lifestyle warning with metronidazole.
Do not drink alcohol. Not a drop. It inhibits the aldehyde dehydrogenase enzyme in your liver. If you drink alcohol while taking metronidazole, you will experience what's called a disulfiram-like reaction. You will get violently, painfully ill with vomiting, flushing, and a pounding headache. It blocks alcohol metabolism completely.
We have covered an immense amount of ground today. From the 1930s dye factories to the modern ICUs fighting superbugs.
It really is a comprehensive tour of chemical warfare.
But looking at all the source material and hearing how quickly these organisms adapt, I'm left with a bit of anxiety. We keep inventing drugs, and they keep inventing resistance. The notes mention persisters bacteria that just go dormant to survive everything. Are we actually winning this war?
That is the big existential question of modern medicine. For 70 years, we've relied on just finding new chemicals. But the discovery pipeline is drying up fast. The bacteria are sharing their resistance plasmids globally. We are already seeing strains of bacteria that are resistant to every single drug we have discussed today. Pan-resistant superbugs.
So what's the future? If we can't out-invent their scissors and pumps.
We have to get smarter. We have to change the rules of engagement. Maybe the answer isn't killing them outright, which triggers that intense evolutionary pressure to mutate. The sources hint at something called quorum sensing inhibition.
Explain that. What is quorum sensing?
Bacteria actually talk to each other. They release tiny chemical signals into their environment to count how many of their own kind are around. When the concentration of that chemical reaches a certain threshold, a quorum, they collectively change their behavior. They suddenly turn on their virulence genes, launch their attack, and release toxins all at once to overwhelm the host.
So they wait until they have an army before they strike.
Exactly. Now imagine if we could create a drug that doesn't kill them, but just jams that specific chemical signal, essentially cutting their communication lines.
They wouldn't know they had an army.
Right. They might just sit there, benign and confused, thinking they're alone. They wouldn't release toxins. And because we aren't slaughtering them, there is much less evolutionary pressure to develop resistance. It would buy our immune system all the time it needs to clear them naturally, peace through jamming the radio rather than bombing the city.
That is a profoundly fascinating thought. It changes the entire paradigm from warfare to diplomacy, or at least espionage. Well, the next time I have to take an antibiotic, I'm going to have a lot more respect for the microscopic, high-stakes battle happening inside me.
As you should. It's a miracle of modern science every time you swallow that pill. Just remember the most important rule. Finish the entire prescription. Don't leave any bacterial survivors behind to learn from the experience and build a better shield.
Very good advice. Thanks for listening to this deep dive.
Stay curious.